Hi, everyone. I'm Sam Alkin, and welcome to our November COVID-19 office hours. We're heading into our second winter with COVID-19, and we're beginning to see cases go back up. And I know a lot of people are saying to themselves, Sam, I thought the vaccines were supposed to get us out of this. And the vaccines are a critical tool to help us get COVID-19 under control. But we are still below 60% vaccination for the total US population. So that means there's roughly 130 million people who aren't vaccinated yet in the US. So there's plenty of people there for the virus to continue spreading. And I know there are a lot of talks about breakthrough cases, but the CDC data clearly shows that vaccines make a difference. If you're unvaccinated, you're 6 times more likely to test positive for COVID-19. And if you're unvaccinated, you are at an 11 times greater risk of dying from COVID-19 if you contract it. So, and now there's only about 16% of the US population that has gotten the booster that's now available to everyone. So the difference between unvaccinated and booster might even be greater than that. So the booster's here. If you haven't gotten it, I highly encourage you to go get it. Now going into the winter, testing is also going to continue to be an important tool. You can't control something, right, if you don't have a way to measure it and verify that the controls that you have in place are effective. That's true for the pandemic. And it's true when you're talking about food safety. But how do you know that the lab performing the test is actually doing it correctly and that the results are valid? Well, that's a great question. And that's the topic for the first part of today's office hours. How do we assess third-party food testing labs to ensure, right, that we can trust what we're getting from them? Now, Martin Wiedmann and Melanie Newman will be leading that discussion, and then we'll open up the floor to ask questions about testing labs, and also feel free to bring any questions you still have around COVID-19. That's what we're always here to talk about. On our panel today, we've got Dr. Olga Padilla-Zakor, who's a professor of food science and director of the Food Venture Center at Cornell University. We've got Gretchen Wall, who's a senior extension associate in the Produce Safety Alliance. We've got Dr. Martin Wiedmann, the Gellert Family Professor in Food Safety. He's the co-director of the New York State Integrated Food Safety Center of Excellence. We've got Dr. Aliosa Trimick, who's an extension associate in our dairy extension program and our COVID-19 resource guru. We've got Dr. Callie Neal joining us. She's a professor of microbial food safety in the Department of Animal and Food Sciences at the University of Delaware, where she explores the transmission of public health and food safety relevant viruses and pathogenic bacteria. We've got Melanie Newman, who is the executive vice president and general counsel of Matrix Sciences International, with expertise specializing in food regulatory compliance and product recall crisis management. We also are lucky today to have Dr. Maria Ishida joining us. She is the director of the Food Laboratory for the New York State Department of Ag and Markets. Since joining in 2015, Dr. Ishida has worked with state, federal, and national associations, such as the Association of Public Health Laboratories and the Association of Food and Drug Officials, to recognize the active participation of laboratories in creating an integrated food safety system. She's going to be a great resource for questions on today's topic. I'm Sam Alkin, an assistant professor here in the Department of Food Science, where I focus on dairy food safety. We have other officials from New York Ag and Markets from both the food safety and dairy divisions on the line. If there are any other questions for them, if you want to ask a question, what you need to do is, if you are on the phone, you need to just press *6 to unmute yourself. Or if you're calling in, you're on the computer, you can just click unmute and ask the question when we get to that point. Or if you feel more comfortable, there's a little chat button at the bottom of the screen. You can click that chat button and type in your question there. You can send it to everyone, or if you want to send it privately to me, then I will read it out to the crew. So think about those questions. I know some of you have already pre-submitted some, and we'll get to those later on in the session. Now, before we start handing it over to Martin, I'm going to hand it over to Al to just give us an update on what are the changes we have going on the Institute for Food Safety website for COVID-19 resources. Take it away, Al.
Thank you, Sam. Yes, so if you visit our COVID-19 page, you will notice that we reduced the number of subpages, and also the content in each of them was reduced. But the pandemic is not over, especially if you check the latest numbers, not very optimistic. So we might still need all the information that was provided on it. So all of the information is archived at the bottom of each subpage. Some of the specific things that we updated: of course, we added the OSHA's emergency temporary standard for COVID-19 vaccination and testing. You will notice that it's not marked as new because this is a bit old news. The new news is that OSHA said that they are taking no steps to implement or enforce this emergency temporary standard for now. Other updates, so we took some subpages away, but we added new ones, so COVID-19 modeling. You can read about the project that we are involved in. So we do a lot of modeling, including modeling how COVID can spread within the food processing environment. For now, you can read about the project we're involved in, but soon we will also add the models that food industry can use. Under tracking COVID-19, we have a tab with peer-reviewed papers where we added one paper where they are showing a little bit of evidence why Delta variant is so much more efficient than the original variants in transmitting from person to person. Under podcasts and webinars, I have to mention that under the same USDA funded grants, there's a regular updates in webinars, and actually at 2 p.m. Eastern time, there will be a third session of webinars where we're gonna update people on progress and all the projects related to COVID and the food industry. So I highly recommend people logging into this. And then virtual office hours, we will post the new dates for our new office hours. And of course, we are adding all the recordings from previous sessions. And we will also add recording from this session. And with this, I think we can continue. Sam, thank you very much.
Thanks, Al. Thanks for the update. You know, we were talking about it earlier. It's kind of like the grocery store. So you got to go back through and go through the aisles and check everything out that's on the website. So thanks. With that, I'm going to hand it over to you, Martin, and bring us up to speed. How are we going to assess these third-party food testing labs?
Thanks, Sam. And first question is why, and what happened to COVID office hours, right? And I think this, because it continues our theme of impacts of COVID-19, not just on, workforce, the more direct impact of COVID-19, but the more indirect impacts. Last office hour, we talked about international supply chains, foreign supply chain verification. Today, I want to talk about labs and et cetera. The labs have been affected by the same COVID issues that affected everyone else, including everyone in the food industry. Supply shortages, same issues. And even if you can get supplies, sometimes what you get is not as good as it used to be. You think about a lab, there's a lot of precision tools there, even simple and cheap stuff that not only do we have less supply, but the supply is sucked up by COVID testing labs. Pipette tips, these little plastic things that need to exactly fit on the equipment in your lab for you to be able to accurately do things. If you get cheap supplies of that, suddenly stuff doesn't work anymore. Let's say that. Labor shortages, same things in the labs as we see in industry and so on, et cetera. So we have sort of anecdotally, seen, and I'll share some case studies, some issues with labs. I'm not saying all labs have problems. I'm not saying it's that widespread, but I think it's a good time to reflect and go back and review your labs, the labs you rely on to assure food safety, and make sure that COVID-19 and other disruptions have not affected them to a point where you might not be able to trust the results as much as you could. So what are we talking about? So the basics is, and what I'm going to convey in you, that it's important to assess both the lab you use and the methods the lab you use. Some of us might fall into a trap saying, hey, as long as I have a lab I trust, how do I decide that? I'm sure they pick the right methods. That might be a little bit too optimistic. I'm going to encourage you today to look past sort of the first level and make sure that you actually convinced yourself that the lab is using the right methods and has the right data to support that the methods are correct for your food. And you need to reassess those basics, both the lab and the methods regularly and when circumstances change. And of course, circumstances have changed substantially over the last year and a half, 2 years. So what should we look at? We start out with the lab. Baseline is your contract lab ISO accredited? ISO says the lab shall define and document how it conforms with the standard. So if it is, there should be some documentation. In addition to ISO, does your lab have regulatory body approvals and audits? Often labs are approved for very specific methods. It might be by New York State Ag Markets in New York for very specific milk tests, et cetera. And Maria can provide us with some input on that at the end of this presentation. In addition to generally assessing the lab, just are they accredited, there are a few key questions to ask. This is not a comprehensive list, but it's really to try to get you started to think about what you should ask. Are they using internal methods or systems audits? How often are audits performed? Do they get external audits? Do they have a lab environmental monitoring programs? Many of us in industry, yeah, we know about Listeria environmental monitoring in our facilities. You probably haven't thought about that labs should do environmental monitoring too, because if you have environmental contamination, you can cross-contaminate your tests. How does the lab prevent cross-contamination overall, and how does it verify that this program works? Does it have specific strains, specific positive control strains that make that easier? How well are the staff trained? Are they retrained? Is the training audited? Or is it simply are we bring in new people and sort of teach them on the job? And again, very relevant right now, turnover in workforce problems, bringing in new people quickly. Often training is one of the first things that falls to the wayside. And we know this from food processing, if we're honest to ourselves. It's the first thing we're gonna do some shortcuts for many, many reasons. Does your lab do the same thing? Does it affect the performance of your lab? Those are the things you wanna look at now and in the future. Lab accreditation, ISO accreditation used to be differentiator, used to be something special. It's a minimum standard. Very similar, think about it in the industry, GFSI audits. That's not, you can't just take a supplier and say, oh, the GFSI audits, I don't need to do anything, I'm good. GFSI audits, approved audits, minimum standard, such as ISO. Just because a lab is ISO certified doesn't mean you should trust it and you don't need to do additional due diligence. There are guidance documents out that can help you with how to select commercial private testing labs. Here is one from FSIS that provides you with some more details. No sense for me regurgitating that here. You can find the document, and maybe Beth even can post it in the chat and take a look at it and use it to help you figure out what to do. Yeah, there are guidances, various guidances out there. And I think the general theme, which is more important, is that the lab at a minimum must have GLP practice audits and participate in some sort of proficiency testing program, and that it needs to use validated testing methods. Okay. And this is one really important thing to keep in mind. And I know many companies, particularly small and medium companies, sometimes forget that. It's not just about the lab, right lab with the right methods. Okay. So the question you need to ask about the methods are the methods validated and verified? And I'm going to explain a little bit more what I mean with this. Any rapid method that you use, that the lab that you select uses, should have AOAC or an equivalent validation. That just shows that the rapid method performs equally well as a standard method that may be laid out in the FDA BAM, Bacteriological Analytical Manual, or in the USDA FSIS MLG, the Method Lab Guidance documents. However, these validation studies typically do not cover all the matrices, all the different types of foods that are submitted by a customer. The kit manufacturers validate their kits based on the foods that they think people want to test and the big markets. They're not going to validate it for some niche product that only a few small companies sell. So when a verification study is needed, when the matrix, the type of food you submit to be tested, was not included in the validation. So you need to make sure the method is validated for your matrix, okay? You probably should have, or at least have seen copies of these verification study results. Do you really know the method will perform up to snuff? And then the other really important thing, is the lab accredited for the method it will do for you? I can have an ISO certified lab that's ISO certified for water testing. It's not ISO certified for food testing, but it offers food tests. There's no guarantee, there's no guarantee ever, but it makes it even more problematic for them doing food testing. So make sure the lab is certified for the methods it uses. Labs can be, and I've seen it, very good about sort of making this very unclear, saying, oh, we are certified by ISO and Agate Markets and FSIS, et cetera, but none of them or actually some of them are not even real certification, none of them relevant to the methods they use for the product they test for you. You need to dig in deeper and assure that the methods are certified and the lab is accredited for the specific method it uses for you. So method validation, let's look at an example to make this a little bit more tangible. So here's a validation claim that you may have. This is a salmonella test. And here it says matrices, hot dogs, frankfurters, beef, raw ground beef, chicken, tilapia, etc. So, it's a little bit from everything, right? A little bit ready to eat meat, a little bit raw, a little bit fish, a little bit juice, a little bit dairy, etc. Looks good. But cheese/mozzarella cheese, okay? You want to use this method to test your raw milk blue cheese. Enough that it was validated in mozzarella cheese? Not a black and white answer, right? Now, raw milk blue cheese, a lot more competitive microflora, Staph aureus in there that could interfere with it, a lot of things. If I'm a blue cheese manufacturer, I probably want to have it verified that this test also works on raw milk blue cheese. Now, we can get more extreme, pet food. Yeah, not validated for. I want to apply this test to pet food. I definitely need verification that this method works for pet food. There are a lot of unique challenges with pet foods. This is what I'm talking about. So just because your test lab says, oh, we're using the BACS salmonella system and it's validated, ask more questions. Validated what? Show me what. And do you really have verification data that works for my product? That's what we're talking about here. It's very clear from different documents that ultimately it is the food manufacturer's responsibility to make sure the method is the right method. You cannot outsource that to the lab. Just because you send the lab blue cheese and they're testing it does not mean necessarily that the method is verified for blue cheese or pet foods. You need to make sure as a food manufacturer that you check on that. And that's one thing you want to do. Now, COVID-19, maybe the lab used a method that was perfectly fine and verified for your food, but supply chain shortages to switch to a different method. Or labor shortages to switch to a method that's easier, requires less labor. Now it's a different method. When did you check the last time whether it changed methods and whether the new method is verified for your food? Something I would do right now if I work in the industry. So why do you not want to trust methods? We've talked about the matrix not being validated or verified, but there are other reasons. Sample sizes not validated. You're testing 375 gram samples, but the method is only validated for 25 gram. We talked about products having weird characteristics, method is not validated. Even if your ingredients come from countries and areas that just have weird specific subtypes, we find this mainly with salmonella. If you import products from parts of Asia, maybe even Africa, the salmonella found there are different. Make sure your methods are validated for that. And remember, when a kit manufacturer verifies and validates that their test detects all salmonella, they don't test all salmonella, they test 100. And in most cases, they get to pick which 100 they test and which 100 they validate for. So if you have complex supply chains, might be time to dig into that a little bit deeper. So what are signs that something is up with your lab? Throughout the ages, but again, particularly relevant during COVID-19. If your turnaround time suddenly became longer or more erratic, ask questions. So you usually get your results in a day, suddenly it becomes 2 days. Even if it's the other way around, it used to be 3 days, now it's 2 days. What changed? What are we doing different? Why do we have a faster or shorter turnaround time? What if it's usually 1 day, but every now and then it's 5 days? What's going on? Ask questions, check in on the lab. If it becomes apparent this lab suddenly uses different test methods, ask questions. Why do we use a different test method? Is it verified for the products that you're testing for me? Is your lab audited to use these methods? Are they certified? Are they ISO approved for these specific methods? They might have been ISO approved for the method, salmonella method they used 2 years ago. They might not be approved for the method they use now. Ask those questions. then some of these warning signs, when you see some of these warning signs. There are definitely other ones, but I think all of us, when we use our common sense, can see what are some of the warning signs of just something not being quite right with the lab. It doesn't mean they're doing a bad job, okay? But it means ask questions and check in with the lab. Should you do a lab visit? One of the questions we got pre-submitted, I would generally say yes. During COVID, height of COVID, you may not be able to do it in person, but ultimately, it is probably a good idea. Again, just like I wouldn't always trust a GFSI audit for a facility, I wouldn't just trust an ISO certification for a lab. If you do it, what should you do? Again, high level, walk through the lab, just take a look at the lab. You can recognize pretty quickly whether they do a good job or not. Find out how your sample moves around. It helps you assess the risk of cross-contamination. Good housekeeping tends to be a good indication of just a well-organized lab. Ask about proficiency performance. Did they do proficiency testing? What are the results? Can you see the results? How does the lab monitor for questionable results? Trends, do we get more questionable results? Do we get more results that are unconfirmed? Are we following up on this? Are we doing root cause analysis? How does the lab assess the potential for lab contamination? How does it prevent it? Is there a log of non-conformances? And again, is there root cause analysis that shows that these issues are dealt with? Now, let's make it practical. A couple of case studies, things that we have seen. One of them, not during COVID, happened pre-COVID. So there was this great lab in California. Everyone, small cheesemakers in particular, loved the lab. It was cheap and the results were always negative, okay? Now, when you go to the web page and read it, it indicates that the lab is ISO approved. But when you then read the more details, it essentially says, we offer food safety testing and we have a strong research program on applying molecular biology tools to identify new cancer treatments. And this is a small single location lab. Looks like maybe 10, 15, 20 people. Okay, if you read between the lines, what does this tell you? What it told me, startup company, trying to make money on new cancer treatments, not successful, investors telling them, guys, or people, we need to have some cash flow here. You have a PCR machine, maybe 2 PCR machines, a couple of people that can run PCRs. How do you create cash flow? Food testing. That's what they probably did. Did food testing. They weren't ISO certified for the method they used. The method wasn't certified by AOC. It was their own homemade PCR method. Happened a few years ago. Lots of negatives. One of the food plants that sent them samples, FDA came in, found a large number of positives, even though all their samples sent to this lab were always negative, and they caused a small outbreak. Why? The lab simply didn't run the right tests and was not qualified to run the test. Critical look at the web page, together with the information you're given, could have told you that if you were critical. Case study 2, this happened during COVID. We have a small lab, reports 2 Salmonella positives for 2 products, same firm, but the 2 products were produced in different facilities, tested at the same time. Both of them Salmonella positive. Yeah, that's weird. Very different products, both Salmonella positive. So we worked with this small company and put in a call into the testing lab and find out they use an immunoassay, but they did not confirm positive results. Despite the fact that what we see on the left side, the simple spec sheet that you can get from the web page clearly says presumptive positive results must, emphasis on must, be confirmed by a cultural reference method, BAM, USD, MLG, or ISO. We asked them about negative controls. Were your negative controls negative? Answer, we didn't run negative controls. Okay, so here are the small food manufacturers, 2 products quarantined because of Salmonella positives. No way for them to assess whether the results are correct because there were no negative controls and no confirmation done. It did get better from there. Lab inquiry response to inquiries via email we got. Our old method for Salmonella involved 3 different augers and broth. Correct, it's a complex method. We cannot confirm that sample with that method. I'm not quite sure what you mean with cannot confirm, but I understand that it's very expensive to maintain these reagents and it's very costly. Phone follow-up call actually revealed exactly that. It's just too expensive. We don't have the money to do this and you don't pay us enough. Essentially exactly the words. I assume you took the same sample that you pick up here and confirmed at Cornell. That will be your results. Most companies just retest the same lots for negative results. Then the lots are good for shipment. I'm not kidding you. This is what an ISO certified lab told a customer. We all know if you have a positive, you can't just take another sample and test negative and the lot is good. It simply is going to be, it's what's expected. It's expected that your second sample is negative because Salmonella almost always is at low levels. There was only one correct, semi-correct thing in here. Consult your inspector. This is a lab that operates in 2021 and receives samples from commercial food processors. So issues are out there. Take a look at your labs. Make sure you're not going to be stuck with some Salmonella positives and have no way to confirm whether these are true positives or not. And lab cross-contamination is not unusual. This is an abstract from a paper. Case of Salmonella cross-contamination of food lab. Conclusion, also cross-contamination food laboratories are rarely reported. Very careful choice of words. Doesn't say rarely occur. It said are rarely reported. Laboratories should always be aware of the possibility of cross-contamination. Results showing us the same serotype in a short time frame from unrelated products should be interpreted carefully. When you talk to Nestle, a large company, started using whole genome sequencing to confirm Salmonella positives, they indicate that 25% of Salmonella positives were traced back to cross-contamination. 1/4 of Salmonella positives were cross-contamination or lab errors. And they were honest about it at an IFP meeting saying, and some of this is our own labs. They didn't just point the finger at our labs. So this is an issue, and you can reduce your chance to being at the wrong end of that stick by evaluating your lab and checking it. So conclusion and action items, ask questions about your lab. Visit the lab if you can. Develop a formal process to approve and regularly review your third-party testing labs. You may have to go and dig deeper into the details of the methods used. Shortcuts on the methods are not unusual. Not confirming when a method requires confirmation. I would hope I could say that it almost never happens. I'm not that confident in it. If it gets too complex, get help if you need help, okay? I wouldn't be a professor if there weren't extra points at the end of today's talk. Question, maybe someone can answer it. What's dry labbing? Anyone want to answer that question? Let's see, anyone in the chat? Come on. Audience participation, please. Aaron says, no idea. Well, reporting results, but not doing the work. Right, okay. 2014, and trust me, it's still out there. Dry labbing is still out there, and there's still a market for it. Dry labbing, a practice whereby research or analysis is claimed to be done, but in reality, the conclusions are guessed at or copied from other sources. Okay, how does this really work? I run a lab. I'm going to get a couple of sponges from environmental monitoring, let's say 20. I could test none of them and report all negative. Some labs, particularly some overseas labs, are known to have done that. And companies that operate overseas know that they need to check their labs. They often send contaminated sponges to make sure there is no dry labbing. The more difficult one is 20 sponges come in. I look at them. 5 of them look really clean. I open the bag, they smell like sanitizer. I'm like, what's the chance of that one being positive for Listeria? Come on, clean sponge, smells like sanitizer. Do you really need to test it? Do you really need to spend $20 worth of reagents? Or should I just call it negative and only test the 15 that look like they might be positive? Profit margin goes up. Again, I'm not implying that a lot of labs do that. I'm definitely not saying all of them do it, but I'm telling you some labs do it. Does it happen in the US? I don't know for sure. Does it happen internationally? Absolutely. Okay. So check your labs. Stuff like that you can probably pick up on if you go to a lab and walk through and sort of be careful enough. So you can trust your lab, but I'm always a big fan of trust but verify, even though it sometimes gets me into trouble. But this is another example of that. So I hope this was useful. I'm happy to answer any questions.